In this video, we will review some of the most important ideas from molecular orbital theory that we will use in organic chemistry too. We have a lot of goals for this video. These goals really are review, so we'll be reviewing these goals from Organic Chemistry 1, and then today we're taking it next level and applying some of the principles that we review to a problem in biology. Have you ever wondered how your eye converts light into a signal to your brain? And no, I have not lost it. This is a chemistry question. We will set it up with a little bit of biology, a little bit of anatomy, but this is a chemistry question. So here's an eyeball, and light hits that eyeball through the pupil, hits the retina, and then something must happen to make a neural signal through the optic nerve to your brain, and then your brain does its magical processing, so you see the image that you see. But at the basic level, light does something to something, some molecule in the retina. All right, that's something in the retina that the light interacts with is a photoreceptor. So you kind of turn the eyeball around here. Here's where light comes through, and then here we have beautiful picture from histology of the retina, the cells that make up the retina. So here's a depiction of the photoreceptor. So something is going to happen in this photoreceptor that's going to send a signal to this bipolar cell, which then sends a signal to a neural ganglion cell, which then sends a signal to your brain. So we are going to zoom in here on the photoreceptor because this is where the organic chemistry is happening. So in the photoreceptor, there are all these little discs, and in these discs are little proteins, and these little proteins in the discs are called rhodopsins. This photoreceptor, of course, is a rod. There are rods and cones in your retina, many more rods. Rods are responsible really for just sensing the presence of light itself. So we're going to think about rhodopsin, a protein that is in these photoreceptor cells in the retina. Okay, here's a rendering of rhodopsin. Now, in rhodopsin, there is a small molecule, and that small molecule is called retinal. You've probably heard of retinal before. From the name, we know that retinal is an aldehyde. Al means aldehyde functional group. So as we'll see, retinal interacts with light and undergoes a change in its conformation, which then causes rhodopsin to undergo a change in its conformation. This big old protein undergo a change in its conformation, and then that's going to interact with another protein, and a bunch of biology stuff is going to happen, and then there's going to be the signal to the brain. So we're going to focus on what happens to retinal, the small molecule, which then in turn causes that change in shape of rhodopsin, which is this protein in these discs inside the rods. And the rods are a type of photoreceptor cell in the retina, right? Excellent. Let's take a look at retinal. Let's look at some skeletal structures here. All right, so this molecule is retinal. Anytime we're encountering an organic molecule, we should stop and analyze its functional groups. We do see an aldehyde functional group and many alkenes. Alkene, 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 alkene. Now we can see around this di-substituted alkene in the middle that the conformation can be described as a cis conformation, to where the two substituents off of the carbons making the alkene are on the same side of the alkene. So this is the cis conformation. They're on this side of the alkene and the hydrogens are on the other side. And it turns out that that cis alkene and the aldehyde functionality are the most important functional groups for the function of retinal and rhodopsin. The first thing that happens is that cis retinal must bond to an amine of the opsin. So it actually covalently bonds to the proteins. So there's some amine side chain, and that is going to bond to the retinal. Now this reaction is part of what's called imine formation, which we will learn about later in the semester. So TBD, don't worry about the chemical reaction going on right there, but we do have the aldehyde plus some amine will generate what's called an imine, and this does so with loss of water. And again, we'll discuss this mechanism later, but for now, you can understand that the retinal covalently bonds to the protein. And you can see this is actually a protonated amine or an iminium ion. Okay, so it's upside down compared to how I drew it over here, but this is a protonated imine. Okay, so look forward to learning that reaction later in the semester. Right now, we're really focusing on this cis-alkene portion of the retinal. So now that the retinal is covalently bonded to the opsin, it is ready to interact with light. So when light comes through the eyeball and reaches the retinal bound to the opsin, that light is absorbed and retinal undergoes a bond rotation. Okay, so here's the light, it's absorbed, and then the cis retinal isomerizes to the trans retinal. So cis to trans, this is an isomerization. Cis and trans retinal have very different conformations. The trans retinal has this linear conformation, whereas the cis retinal is not linear, right? It has this bend where that cis alkene is located. This new shape of retinal changes the shape of the opsin protein. And that change in shape of the opsin protein results in a signal being sent to the brain. So recall that we were looking at the picture of rhodopsin. We said, so retinal, here it is in its cis conformation. It absorbs light. It turns into the linear trans conformation. That causes a change in shape here in the rhodopsin. Just a quick aside. So I was calling it opsin on the previous page. These proteins are called opsins. This is a rhodopsin because it's in a rod. So rhodopsin, opsin, I'm referring to the same thing. So the rhodopsin changes shape, and then here, and then you hear our friends in biology say all the time, structure and function, structure and function. So when it changes its shape, it has a new structure. There's another protein that senses that, and then as I said, some biology happens, and then the signal is sent to the brain. That biology is called the phototransduction cascade, and I'm sure many of you will learn all about it one day. But that day is not today. Cool. All right, now we know how light is converted to a signal in the brain. Basically, we know the chemistry, right? So let's just discuss what we learned just now. So what is the relationship between cis and trans retinal? I'd also like you to ponder this question. So do cis and trans alkenes often interconvert quickly at room temperature in the absence of light? Cis and trans retinal are isomers. They have the same molecular formula, C20H28O, but they're not the same. Now, they have the same connectivity of atoms. The atoms are bonded in the same order, so they are not constitutional isomers. They differ in how those atoms are arranged in space. So they are stereoisomers. Stereo, space. Well, if they are stereoisomers, and clearly they are, you can look at them and you can see the atoms are arranged differently in space. They're either conformational or configurational. Conformational isomers can be interconverted with simple bond rotation without breaking any bonds. Are cis and trans retinal conformational isomers? To interconvert between configurational isomers requires breaking a bond. So which do you think it is? Conformational or configurational? Yeah, cis and trans alkenes and cis and trans retinal are configurational isomers. And specifically, they are diastereomers. They are configurational stereoisomers that are not mirror image isomers, right? Enantiomers are non-superimposable mirror image isomers. This is not the case. If we tried to superimpose, meaning if I laid the trans retinal on top of the cis retinal, the atoms would not line up, right? These R groups would not line up. So they're not superimposable, and they're also not mirror images, right? This is not a mirror plane that reflects the trans onto the cis. So these are diastereomers. They're stereoisomers that are not configurational stereoisomers that are not enantiomers. So do cis and trans retinal interconvert quickly at room temperature? And then I have this in the absence of light because this problem involves light. So what do you think? Well, let's think about what you may have answered. So you may know that trans alkenes are more stable than cis alkenes. So it is true that this trans alkene, I'm at the bottom of the slide, is more stable, lower energy than the corresponding cis alkene. And that is due to steric repulsion, so repulsion of the electron clouds in the cis alkene. However, the trans is not low enough energy that there's interconversion at room temperature. There's no driving force for that. So cis and trans alkenes do not rapidly interconvert at room temperature. And this is because to interconvert between these two isomers requires breaking a bond. Remember the definition of configurational isomers. Bonds must be broken to interconvert the two isomers. What bond is broken when the cis isomer is converted to the trans? Yes, the pi bond. The pi bond would be broken. And that would require energy that is not available at room temperature. Great. Well, cis alkenes don't rapidly interconvert to trans alkenes at room temperature. That is because the pi bond will be broken. Well, how do we know that? Can I just say, oh, the pi bond's broken? How can we explain why cis and trans alkenes don't interconvert quickly at room temperature? How can we explain this with valence bond theory or molecular orbital theory at a level that is appropriate for organic chemistry too? Give it a try. Pause here and try to explain why cis and trans alkenes do not interconvert at room temperature. Show what's going on with the pi bond if in fact we were correct and the pi bond must be broken to interconvert these two molecules. Then I've rotated the alkene 90 degrees, the cis alkene. So the R groups are pointing out at us. And just, the R groups here's an R group, here's an R group. Much easier to abbreviate these as R than to draw the entire molecule again. Definitely not going to do that. So in the cis right now, here's the p orbitals, nice and aligned to make the pi bond. These p orbitals are making the pi bond. And to make that pi bond, they must be aligned. Now, right now, the two R groups are coming out at us, right? They're on the same side, they're cis. To convert to the trans, there must be bond rotation around this carbon-carbon bond to get that R group going into the page on the other side. And if you have a model kit, you can try to rotate around this bond, this pi bond. What's going to happen? What happens when you try? And it probably requires a lot of force, and if you finally get it to rotate, you're going to break the bond. Okay, so here we are to the trans. You can see now the R groups, one's coming out at us and one's going into the board, so that the R groups are trans, like they are here in this rotated picture. Okay, great. I mean, it looks simple, right? We just rotate around the central carbon-carbon bond, and then the cis is converted to the trans. Not so simple. Let's imagine the bond rotation. I'm going to rotate this carbon atom. Let's imagine grabbing this R group, and now we're going to rotate it up. And when we rotate this R group up in space, so that eventually we rotate it 180 degrees to make the trans isomer, we go through this intermediate conformation where we've just rotated the R group up 90 degrees, so rotated 90 degrees around this bond. And now what we're looking at here, this is a bird's eye view of the p orbital. So that p orbital there, this one, used to be aligned with the other p orbital. Now these two p orbitals are perpendicular to one another. So if p orbitals must be aligned to make a bond, These two p orbitals are perpendicular. They're at a 90-degree angle to one another. What's happened? Yes, that bond is broken. So although if we rotate through this conformation, the cis can be converted to the trans, the bond is broken in this intermediate conformation. The pi bond is no longer there because these p orbitals are not aligned. They must be aligned to make a bond. If we rotate this R group to get it over here, make the trans conformation, it requires this intermediate conformation where the p orbitals are perpendicular, not aligned, thus the bond is broken. And that requires energy, right? That requires energy. And that energy is not available at room temperature. It's available at super high temperatures, but not at room temperature. And why am I mentioning room temperature? Well, the problem at hand takes place in your eyeball. So, okay, sure, your eyeball is not at room temperature, but it is at body temperature. And there is also not enough energy at body temperature for this transformation from the cis to trans retinal to occur. Now, believe it or not, this is our big question for today. The question is, how does light cause this transformation from cis to trans retinal at a level appropriate for organic chemistry 2 students? If you're asking, well, what is a level appropriate for organic chemistry 2 students? This level right here, based on orbitals and electrons and chemical principles that we have learned thus far through general chemistry and organic chemistry. Can we solve this problem? Of course we can, but we might have to think kind of hard. So let's take a moment and stop and think. We're going to write down what we already know that might help us solve this problem. We're going to write down what we might need to know in order to solve this problem. Then we're going to go learn this stuff. We're going to research this stuff. Then we're going to come back and we're going to write down what we learned. And then we're going to solve the problem. We are going to explain on a molecular level how light can cause the transformation from cis to trans retinal. So let's go. Write down what you know and what you need to know. You may have written down that alkene isomerization requires energy. You may have written down that alkene isomerization breaks the pi bond. You may have written down that trans alkenes are lower energy than cis alkenes. Now, remember, we don't exactly know what is going to be relevant about what we know, but if we think it might be helpful, let's just write it down. We know that light is involved. It's light that promotes the isomerization of the cis to trans. We know that cis and trans alkenes are diastereomers. You may have written down many other things. What do we need to know? Well, we need to know how light interacts with the alkene. And it's possible that some of you already know how light interacts with atoms and molecules, and that could be in your what you already know. All right, so maybe you might not know exactly how light interacts with the alkene, but you do know that light can excite electrons. Maybe that's helpful. We know that from general chemistry. So what do we need to know? Well, how does that apply to the alkene? Maybe can the electrons in the alkene be excited? We know they can be excited in atoms. Can they be excited in molecules? And what happens to them? All right, there's many things that you can write down here. You may have not written down these exact items, and that's fine. We're all different. Let's get started, though, working on what we need to know. And a lot of these questions can be answered by, yes, molecular orbital theory. Now, before we can think about the molecular orbitals of an alkene, especially a big alkene like retinal, let's take a moment and think about the simplest molecule, which is hydrogen. It has 2 protons and 2 electrons. Fewest parts, simplest molecules. Blue structure for the hydrogen molecule looks like this. There are 2 shared electrons between those 2 hydrogen molecules. We can describe the location and the energy of the electrons in this molecule. The electrons are in a sigma orbital, most likely to be located between the two nuclei. That's where there's the greatest probability of the electron existing. And those two electrons are lower energy there than they were in the atom, right? That's why bonding occurs. The electrons are lower energy between the two atoms than they were on the individual atoms before they bonded. Awesome. Well, in valence bond theory, we certainly do show that the overlap of atomic orbitals forms a single covalent bond. However, the rules of quantum mechanics require that the number of orbitals be conserved. When two orbitals are combined, two new combinations must result. So when a 1s orbital on one hydrogen is combined with a 1s orbital on another hydrogen, there is a sigma orbital formed. We already drew that, described that. But there is also another orbital formed. Two atomic orbitals, when they combine, must generate two molecular orbitals. Those are the rules. We will now use hydrogen as an example. Now I'm going a bit quickly here. If you do need a slower, more in-depth review, please see the full video from the Organic Chemistry 1 lecture series. It's about video 8, I believe. There are two possibilities for what can happen when the two 1s orbitals combine. They can add in phase to generate that sigma orbital. Recall that orbitals are described by wave functions. I'll just draw some simple waves here. So I've just drawn generic waves here. Of course, if we're being super technical, there's not a node in the 1s orbital, so maybe I should have just drawn it like that. Let's not worry too much about it. We have two waves. These are in phase. When we take this one and overlap it with the other one, they would constructively interfere, meaning add up, and we would get a bigger wave. All right, so that's what's happening in between the two nuclei. Where those two orbitals come and overlap, there's constructive interference. Now, the second possibility is that the two waves are added out of phase. We can represent that by showing different shading, right? Shaded and unshaded show the two different possible phases, positive and negative, of the wave function. So we could also bring those two 1s orbitals together out of phase, which I'm going to represent with these super simple waves like this. Okay, so this would be out of phase. So if I took this wave and overlaid it on that one, they would destructively interfere, cancel out. And that is in fact what will happen between these two nuclei when we bring them together. Those two orbitals are out of phase, so they will destructively interfere in between the two nuclei to generate what is called a sigma star orbital. So we can show the new node. This is where the destructive interference occurred, right? Nodes are regions of zero electron density. There is no electron density between those two nuclei. Now, it looks like the two orbitals are like leaning away from each other. No, they canceled out. There's a node there. There is zero electron density between the two nuclei in this orbital, which is called sigma star. It's also called an anti-bonding orbital. So when these two orbitals are combined, a bonding orbital is formed, sigma, where the two atomic orbitals constructively interfered between the two nuclei, and there's electron density between those two nuclei now in this orbital, and the anti-bonding orbital, where the two atomic orbitals actually canceled out in between the two nuclei. So there's a new node formed, nodes being regions of no electron density. We can represent these orbitals on a molecular orbital energy diagram. We'll put the atomic orbitals, AO, on the side. So here's one of the 1s orbitals. Here's the other, and the new molecular orbitals that are formed in the middle. So when these two 1s orbitals come together, they make a lower energy bonding orbital and a higher energy antibonding orbital. The bonding orbital is lower in energy, about the same amount that the antibonding orbital is raised in energy. Where do the electrons go? Well, the two electrons go in the lowest energy orbital, which is the bonding orbital. Overall, the electrons are lower energy than they were in the atoms, and that is why the bond is formed. Electrons become lower energy. and they serve as the glue to hold those two atoms together in the hydrogen molecule. All right, be sure that you have a handle on this before we use molecular orbital theory to explain why helium-2 does not exist. And again, one more time, if you need more details or slower explanation, more examples, go to the full molecular orbital lecture from Organic Chemistry 1. Okay, we could draw both the helium and the hydrogen molecules, and we're going to use molecular orbital theory to explain why helium doesn't exist. So no, we're not going to say, oh, but helium already has a full duet. No, we're going to use molecular orbital theory to explain it. The 1s orbitals on hydrogen or helium can come together in space and overlap to make a lower energy orbital when they add in phase, and a higher energy orbital when they add out of phase, just like we showed on the previous slide. Adding in phase results in the sigma bonding orbital. Adding out of phase results in the sigma star anti-bonding orbital. Both of these orbitals are made when either the two hydrogen atoms or the two helium atoms overlap in space. Now that we have the molecular orbitals, we can put electrons into them. Each hydrogen atom starts out with 1 electron. Each helium atom starts out with 2 electrons. The 2 total electrons for the hydrogen atom go into the lowest energy molecular orbital. The 4 electrons of the helium molecule, 2 go into the bonding orbital and 2 go into the antibonding orbital. All right, now we can calculate something called bond order. Bond order approximates what in valence bond theory we would call a single or a double or a triple bond, but uses molecular orbital theory to determine. Bond order is 1/2 the total number of electrons in bonding orbitals minus the total number of electrons in anti-bonding orbitals. Let's calculate this for the hydrogen molecule first. So the bond order for the hydrogen molecule would be 1/2 the total number of electrons in bonding orbitals, which is 2, minus the total number of electrons in antibonding orbitals, which is 0. 1/2 of 2 is 1. So just as valence bond theory suggested, molecular orbital theory predicts a single bond between those two hydrogen atoms. Let's go over to the helium molecule here. The bond order would be 1/2 the number of electrons in bonding orbitals, which is 2, minus the total number of electrons in antibonding orbitals, also 2. 1/2 of 0 is, of course, 0. So molecular orbital theory predicts no bond, no bonding interaction between these two helium atoms. Although 2 electrons were stabilized by populating that low energy antibonding orbital, the other 2 electrons were destabilized at higher energy due to populating the antibonding orbital. So overall, the effect of the bonding orbital is canceled out by the higher energy effect of the antibonding orbital. Okay, what have we learned here? We learned that filling an antibonding orbital breaks a bonding interaction. That was fun. I hope we can use this for the problem we're trying to solve. All right, at this point, there are several common questions that pop up, and again, I go through all of these questions in detail in the molecular orbital theory lesson from the Organic Chemistry 1 lectures. However, I will briefly address each of these questions here as well. So you might be wondering, why do both bonding and antibonding molecular orbitals form from the combination of atomic orbitals? Well, remember that the model for orbitals are wave functions, and wave functions can have 2 different phases, positive or negative. So when you combine 2 orbitals, there are 2 possibilities that the 2 orbitals will have the same phase as represented here, and constructively interfere or overlap, or that the two orbitals will have different phases, as represented here. Overlap of those two orbitals in different phases will result in destructive interference, or the two orbitals canceling out. Constructive interference leads to a bonding orbital, and destructive interference will lead to an antibonding orbital. Speaking of antibonding orbitals, what is an antibonding orbital anyway? We'll consider the bonding and antibonding orbitals discussed today. And the easiest way to do this is to think about the effect of an electron on the nuclei. So these points are the nuclei. In the bonding orbital, the most likely place for the electron to be is between the nuclei. In the antibonding orbital, the most likely place for the electron to be is outside. We can represent those two scenarios like this. Let's look at the bonding orbital first. In the bonding orbital, the electron being between the two nuclei serves to pull those two nuclei towards itself. Pulling the nuclei together. That is the definition of a bond. And obviously, I am simplifying it a bit. There certainly are repulsive interactions between the nuclei as well, but we're not worried about that right now. We're worried about what an electron does in each type of orbital. Now let's consider the other scenario, an electron in an antibonding orbital. There is a node between the nuclei in an antibonding orbital where those two orbitals, the atomic orbitals, when they came from together with different phases, destructively interfered. So the most likely place for the electron to be is on the outside of the bond, such as right here. What effect does that electron have on the two nuclei? Well, it really strongly pulls this nucleus towards itself, and it kind of pulls that one. But there's all this repulsion between the two nuclei, and these unequal attractive forces between each nucleus and the electron serve to pull the nuclei apart. Well, that's the opposite of a bond, aka antibond. All right. Well, now we understand a little bit better what an antibonding orbital is. Why do they matter? You said they're usually empty. Well, they can be filled. They can accept electrons just like any orbital can accept electrons. And then what happens? What do we see happen for the helium molecule? Yeah, filling the antibonding orbital resulted in no bond. So filling antibonding orbitals breaks bonds. Bond breaking and bond making, well, that sounds like chemical reactivity, and that is what we are studying as chemists, and particularly as organic chemists. Okay, so now that we can draw simple molecular orbital diagrams for small molecules like hydrogen, let's apply this to an organic molecule, and we will look at ethane, which is C2H4, more commonly referred to by the name ethylene. Go ahead and give that a shot, especially if you did the warm-up problems for this lesson. Let's start with the carbons. So there's a carbon-carbon single bond. That means that there's one sigma Cc bond and one sigma Cc orbital and one pi Cc. We know that if a sigma Cc was formed, so was a sigma star orbital. If a pi orbital was formed, so was a pi orbital. So even though I expect these antibonding orbitals to be empty, they do exist. Now to the four sigma CH bonds. Those bonds have electrons in sigma CH orbitals. If there are four sigma CH orbitals, there are four sigma star CH orbitals. The next thing that we do is we order the orbitals on our energy diagram and from lowest energy to highest energy. Then we will populate them with electrons. First things first. Quick reminder, we know that bonding orbitals are the lowest energy, then non-bonding orbitals, such as atomic orbitals, right? We started out at atomic orbitals, bonding were lower energy, antibonding were higher energy. All right, and then antibonding pi star is lower energy than sigma star. Okay, so our lowest energy orbitals will be sigma Cc and sigma Ch orbitals. Now, sigma Cc will be just a tiny bit lower energy than sigma Ch. Recall that within a certain type of orbital, such as sigma, we have sigma Cc versus sigma Ch. Those made from more electronegative atoms will be lower energy. That is the convention that we will be using in this model of molecular orbital theory. Carbon being slightly more electronegative than hydrogen results in sigma Cc being slightly lower energy than sigma Ch. Next, the pi Cc orbital. After that, pi star, because there are no non-bonding electrons in ethylene, right? There are no lone pairs. Then after that, sigma star, and again, sigma star Cc is expected to be slightly lower energy than sigma star Ch. So we had these two principles we used for organizing the orbitals. Bonding is the lowest energy, where sigma bonds are lower energy than pi bonding orbitals, then non-bonding, then pi star, then sigma star. And then if we had orbitals of the same type made from different atoms, those made from more electronegative atoms, we made lower energy. All right, let's put the electrons into the proper orbitals. So for ethylene, there are 2, 4, 6, 8, 10, 12 electrons, which we can get from the Lewis structure or from the molecular formula using the valence electrons of each neutral atom. So 2, 4, 6, 8, 10, 12. Now we want to check to see that bonding orbitals are filled and anti-bonding orbitals are typically empty. You should be able to draw the cartoon molecular orbital pictures for each of these orbitals, just like we did for hydrogen. We drew the sigma and sigma star orbitals for hydrogen. You should be able to do that for each of the bonds in ethylene. Again, to spend a lot of time doing that, you can review the full molecular orbital lecture from Organic Chemistry 1. I will quickly, however, help you with the sigma Cc interaction. So each of the carbons in ethylene is sp2 hybridized. Therefore, each of those carbons will bond to the other using an sp2 orbital. Those two orbitals could come together in phase and constructively interfere, resulting in this sigma Cc bonding orbital. Conversely, these orbitals could come together out of phase, and instead of overlapping in between the two atoms, they will destructively interfere and cancel out. So where this shaded lobe interacts with this unshaded lobe, it's like the positive phase interacting with the negative phase of the wave. Now in this picture, it looks like the big lobes and the small lobes have traded places, but that is not what has happened. What has happened is that these two big lobes canceled out in between the two nuclei, and there's a new node there. So these represent the cartoon molecular orbitals for sigma star Cc and for sigma Cc. Sigma Cc. Let's go ahead now and show the pi and pi star orbitals as well. The two orbitals that make up that pi bond, the two p orbitals, can come together in phase where the two shaded lobes overlap and the two unshaded lobes overlap. This will form a pi orbital. We can show this pi bonding orbital two ways. We can just line up the orbitals in phase, and every chemist knows, oh yeah, she means a pi orbital. Or you can draw the hamburger. All right, either way. So here's our pi Cc bonding orbital, where the two atomic orbitals have constructively interfered between the two nuclei. Again, conversely, those two p orbitals can be brought together out of phase, where the shaded lobe interacts with the unshaded lobe on top, and the unshaded lobe interacts with the shaded lobe, and they're going to cancel out in between the two nuclei, which will generate a pi star orbital. Pi star can be represented by just lining up the two p orbitals out of phase and drawing a node in between them. Or you can draw the butterfly, which attempts to show how the two orbitals have canceled out in between the two nuclei. So we can add the node there. Either representation is fine. These two cartoon molecular orbitals represent the pi and pi star CC molecular orbitals. Pi star CC, pi star CC, and pi CC. Excellent. So you can try the sigma star CH and the sigma CH on your own if you like, or again, consult the more in-depth video. Well, that was a lot. Let's check in with our what we need to know list to see if we've learned anything helpful for solving the problem at hand, which is how light can cause the transformation from cis to transretinal, which we know involves bond breaking. And we know there's not enough energy available at either room or body temperature to affect this bond breaking. How is light doing it? What we need to know. Looks like we have a lot of questions about how electrons in the molecule can be excited, what happens to the electrons, and how light interacts with the alkene. You know, something that we probably should have had in our what you need to know column related to these questions is just in what orbitals are the electrons in the alkene? And then we can ask, you know, what happens to these electrons? So there's a few questions left we need to answer. Can electrons in a molecule be excited, and how does light interact with the alkene? I think that our MO diagram will help with that. So let's review now. What happens when electrons interact with light? All right, so what else do we need to know? Well, let's go back to general chemistry and let's think about what happens when an atom absorbs light. So an atom has all these atomic orbitals. It has electrons in the atomic orbitals. And what happens when an atom absorbs light? If you can't remember, you can go look it up. So when an atom absorbs light, that corresponds to the energy difference between two energy levels. An electron is excited. It is promoted to the next highest energy level. Okay, here's that electron that is promoted. And of course, for the atom, we know this electron can relax back down and we can absorb light being emitted from the atom. A few vocabulary terms. The lowest energy state of the atom is called the ground state. All of the electrons are in the lowest energy orbitals. And then the excited state represents where the electron has been promoted to a higher energy orbital. Now, what happens, do you think, when a molecule absorbs light? Yes, something similar. Instead of atomic orbitals, the molecule has molecular orbitals. Those lowest energy molecular orbitals are populated with electrons in the ground state. The molecule has a ground state as well. And an electron in the highest energy occupied or filled molecular orbital, aka the HOMO, will be promoted to the LUMO, which is the lowest energy empty or unfilled orbital. So unoccupied, but I'm writing unfilled. All right, molecular orbital. As long as that light has an energy equal to the difference in energy between those two orbitals. That yields the excited state of the molecule. So basically the same thing happens to a molecule when it absorbs light as to an atom. The exact orbitals are a bit different. The changes in energy are a bit different, but it's the same process. An electron can be excited by the light. So let's take a moment now and write down what we have learned because we're about to attempt to solve this problem. So I'm not going to do this for you. If you want to make the most of this lesson, you've spent all this time listening to it, write down what you've learned. It doesn't matter whether or not you're going to end up using it, but write it down, especially if it relates to some of these questions. Okay, we're going to move on. And we have what we've learned here. So let's try again. Explain how light causes the transformation from cis to trans retinal. You need to do this. Write down your explanation, and then you'll check with me. Let's start by using what we've learned. So the answers to the questions that we asked. What does light do to molecules? Well, it promotes an electron. Follow up, what orbitals are the electrons in retinal in? Now, you might say, okay, do I need to draw a complete molecular orbital diagram for retinal? You could if we were maybe in physical chemistry, or if we were in graduate school, maybe we would draw a full molecular orbital for retinal. But we don't need to do that. We can try to pick a simple model to describe what's going on. Just like we started with looking at the hydrogen molecule when we needed to think about molecular orbitals. For retinal, let's just choose a model that describes where the action is. So the action's at this pi bond. So I think we can use ethylene as our model. Let's try it anyways. And luckily for us, we've already drawn out the full molecular orbital diagram for ethene. So let's go get that, paste it here. All right, here it is. The next time you see it, I'm going to have cleaned it up a bit, but I am taking it from this slide. Here's our MO diagram of ethene. We know where the electrons are. Now let's imagine that light hits ethene, which is our model for cis-retinal. So let's imagine it hits cis-retinal. We know that an electron in the highest energy filled or occupied molecular orbital will be excited. It will be promoted up into the LUMO. This represents the lowest energy transition for this molecule. So that electron will be promoted to yield the excited state of ethene, which we're using as a model of the excited state of cis-retinal. Well, what do we notice here? There's an electron in pi, and there's an electron in pi star. What does this mean? What is a consequence of an electron being promoted from pi to pi star? Is anybody really excited right now? Did you just realize the punchline? Yes, when the electron is promoted to pi star, the pi bond is broken. Eureka! The pi bond is broken. Right, think about the bond order. Remember bond order? You wondering why I was going over that? Well, this is why. Just thinking about the bond order of the pi bond. There's 1/2 of one electron in the bonding minus 1/2 one electron in the antibonding. What's 1/2 of 0? 0. There's no bond. It's broken. You can't see me, but my arms are raised in victory. We have figured it out. Let's summarize our solution. So I've rewritten out these diagrams in an even more simplified form, just considering the pi orbitals. So let's work through this together. We start out with cis-retinal in the ground state. Cis-retinal absorbs light and an electron is promoted from pi to pi star. In the excited state of cis-retinal, which is indicated by this star, this typically means excited state, the pi bond is effectively broken by having one electron in the pi orbital and one in the pi star. We can represent cis-retinal as this diradical species where there is no longer a pi bond. And then now this species can freely undergo rotation around this single bond to yield the lower energy trans conformation of the two alkyl substituents of the carbons making this double bond of retinal. We mentioned early on in our brainstorming that transalkenes are lower energy than cisalkenes. So once the pi bond is broken and free rotation is allowed, this trans relationship of the alkyl groups is lower energy. In the cis conformation, these two alkyl groups are eclipsed, and in the trans conformation, those two alkyl groups are anti to one another. From the excited state of transretinal, the electron from pi star can relax back into pi to yield ground state transretinal, as represented here, right? That was the goal. How is cisretinal converted into transretinal? And then, as we discussed 45 minutes ago, transretinal causes this conformational change in the rhodopsin, which causes a change in another protein, which does the phototransduction cascade, and bang, your brain gets a neural signal that light is present. I suspect that some of you did not believe me, but as promised, this was a chemistry problem. And I had a lot of fun solving it with you. But it wasn't all about having fun, and it wasn't all about looking at biology. We reviewed several really important both chemical principles and processes that we'll be using to problem solve in the coming weeks. So take some time and reflect about how we solve the problem, really focusing on process, and then also what chemical principles we learned. And also, I hope I showed you why organic chemistry is required for your field of study. It's not just about memorizing a bunch of reactions. Later on in your studies, it is really helpful to actually understand how and why these phenomena in biology occur. And you'll have the tools. Wonderful. So we'll always end our class by summarizing. So be sure you write down what you learned today and what questions you have, and send me an email or comment on the video and I'll try to answer your questions. Be sure that you can achieve all of the learning goals for today. I did try to hit all of these review goals, but again, you can check out the full lesson from Organic Chemistry 1 if you would like. We have fully described how light interacts with the electrons of molecules, same way it does with atoms. It can promote an electron, given that there is a transition between orbitals with the same energy as the energy of light. And then finally, I hope you had as much fun as me applying the basic principles of MO theory to biological phenomena. Again, it was about having fun, but it was also about the basics of MO theory, because we will be continuing to apply these to chemical and biological problems in the coming weeks. There are some practice problems. This problem is from the warm-up, and then you should draw the full MO diagram and identify the HOMO and LUMO of acetonitrile. And then there's one more problem, the complete MO diagram of acetylene. And again, once you've drawn that, you should identify the HOMO and LUMO. Okay, as always, time to practice.